figured out where am I. Sorry, you can see my slides?
Yes, thanks.
Okay, very good. Okay, good morning, everyone. Thanks so much for being here, and thanks to Dion and to the CDFCP for hosting this webinar. Just make sure my slides actually advance here. Yes, okay, so the research I am presenting today centers around Galiano Island, an island that is centrally situated in the Salish Sea in the territories of Halkamenitnam and Strait Salish peoples. With this presentation, I want to begin by acknowledging that Indigenous people have been stewarding these lands and waters since time immemorial and have their own knowledge of the profound changes that have taken place here since colonial times. So the regional climate may be described as semi-Mediterranean with mild, wet winters and warm, dry summers. And you probably know what this looks like in summer months with the seasonal drought that we experience, which is a critical environmental stressor for plant and insect communities and something that we expect to worsen under future climate scenarios. Like many of the Gulf Islands, Galiano Island has been subject to an intensive history of forestry, agriculture, and land conversion, though today it remains relatively intact compared to other nearby islands. This history of post-colonial landscape change has radically impacted these ecosystems, resulting in altered predator-prey dynamics and changes in the composition and structure of ecological communities. It's against this background of climate and landscape change looms the problem of shifting baselines. Ecological communities are changing, and these changes happen often without our even noticing due to a lack of historical baseline information. With COP15, Canada has made a commitment to monitoring biodiversity in an effort to curb its loss, and yet to date, realistically, we lack the framework required to reliably detect biodiversity change. So today I'm going to speak to the potential for community science to help tackle this challenge by implementing systems for tracking biodiversity change at the local scale, which can complement initiatives happening at broader provincial and national scales to develop a biodiversity observation network in Canada. We'll look at two case studies. One, a recently published study documenting historical change in the island bumblebee community, and the other, an ongoing study looking at historical change in the vascular plant community. These studies both draw upon data generated through the Biodiversity Galiano project, a systematic effort to synthesize a baseline biodiversity dataset and to meaningfully engage our community in the process. The BioGaliano project was founded in 2016, so it's been going on now for just about 8 years. The BioGaliano project begins with the synthesis of historical biodiversity data, and then we incorporate community science data, which we then compare with our historical baseline information. Comparing these two sources of information, the outcomes are quite simple, but very useful. First, you have the list of all species that are historically reported for Galiano. Then, as we continue our search efforts, we have the growing list of confirmed reports, that is, species historically reported that are subsequently documented by project contributors. We get the list of any new species that may turn up. And importantly, we get the list of unconfirmed historical reports. Now, again, this may seem quite simple, and yet this is really the fundamental information required to begin the work of detecting biodiversity change. Over time, as this information accumulates, one can obtain a rich overview of the biodiversity present in an area. You learn a lot about the species present, but also about what's absent. You begin to get a sense for the gaps and the groups that might be undersampled, as well as a great deal of information that is useful to guide further research. To date, we've documented over 4,000 species on Galiano Island. Historical records comprise about 60% of the total known species, with the remaining 40% coming from species reported new for the island since the project began in 2016. So far, we've confirmed about 57% of the historically reported biodiversity, which means there are just over 1,000 species historically reported for the island that have not yet been detected after 7.5 years of organized search efforts. So just a quick overview of what we know about our local arthropod diversity. This is a large phylum that includes insects such as bumblebees. There are about 1,500 species reported to the island so far. This tally, 640 species reports are based on historical records, of which we've documented 229 since the project began, confirming 36% of the reported diversity. And we've also added 859 species to the list. And still at large, things that are unconfirmed but historically reported are about 400 species. This is a gap that we aim to close as we continue our search efforts. For vascular plants, so far we've documented just over 800 species. About 3/4 of these species are historically reported, of which we've so far confirmed 500, that is 80% of the historically reported diversity. And about 200 species have been added new to the list. And finally, still at large are about 100 species. So having established these baselines, I'll just now go over how we've used this information to detect historical change in these communities. First, we'll consider the bumblebees. And from the outset, I'll just note, I've really had to abbreviate these case studies to keep within the time limit. So if you're interested in methods, et cetera, we can discuss that during the question period, or else you can have a look at this paper published in Northwest Science. So in this study, we combined several sources of biodiversity data to establish evidence for historical change in the community, including voucher specimens accessioned at the Royal BC Museum and the BD Biodiversity Museum, iNaturalist observations, and then bumblebees sampled using blue vein traps. So again, I'll have to gloss over many of the details of these studies, but here's a quick overview showing the distribution of historical data, iNaturalist observations, and the sites I sampled in 2018, those white dots there. And you can see they're pretty well distributed. The sampling effort was quite intensive over 5 months during 2018 and all over the island. So from these 3 sources of data, I generated rarefaction curves, which they estimate the expected number of species in a sample as a function of the total number of individuals sampled. So you have the total number on the bottom, and then on the y-axis you have the number of species. So this curve for the historical data shows that there were at least 10 bumblebee species historically occurring on Galiano Island. Comparing this rarefaction curve with the curve generated based on iNaturalist observations, using a null model test, we found that there was a significant difference with only 6 species occurring in the contemporary community. We also showed the same curve, basically, a significant difference between the historical data and the data generated based on blue vein samples. So this much more intensive sampling effort also showed a significant change had taken place. Importantly, the curves generated from iNat and blue vein sampling both converged on the same estimate of species richness, demonstrating that both sampling methods were sufficient to estimate richness in the present community and to detect change with reference to the historical baseline. So the story that emerges from the study is fairly dramatic, and Pam was saying not necessarily a happy story, but what we've seen is the apparent loss of half of the historical bumblebee diversity, as well as the recent arrival of a new species. And importantly, this study was the first to establish robust evidence for the use of community science platforms like iNaturalist as a non-lethal sampling method to detect historical change in bumblebee communities. So quickly, there are, of course, some limitations to consider here, and I'll come back around to considering some of these in the conclusion. But first, of course, you need to have adequate historical baseline data to take this approach. In our case, we're looking at a relatively small species pool that's just 10 bumblebee species max, historically known to occur. Galiano Island is a relatively small study area. And I was able to show in my research that bumblebees are really ubiquitous in the environment. So they're not as problematic as other groups like vascular plants. Moving on to vascular plants, our next case study. Here's a quick overview of historical search effort. On the left, you can see the historical data from 1948 to 2014. And on the right, all the additional data that's been generated since the project began in 2016. By 2020, we had documented about 80% of the native plant diversity. From the list of historically reported species that hadn't been observed, we narrowed down 14 candidate species that we deemed to be at risk of extirpation, and we set out in a concerted effort to detect them through systematic surveys of historical and potential habitat. Some of these species are ranked as endangered in Canada and so are very high priorities for conservation. Others, however, are not necessarily considered rare at the national or provincial level, but nevertheless appear to be at risk of extirpation at the scale of Galiano Island. Altogether, from 2020 to 2023, we logged hundreds of hours of search effort targeting these species. And here you can see an overview of search effort on Mount Soodle, which is an important site for several of these plants. And after 3 years of targeted search effort, we have been able to detect half of the species on our list. So now it remains for us to consider the probability that the remaining species have been extirpated. So this case study is much more difficult than the case of Galiano Island's bumblebees because there are many more factors to consider in evaluating the likelihood of change. For example, unlike bumblebees, which I was able to show are ubiquitous in the environment, that is, everywhere I sampled, I found the same species, vascular plants tend to be much more restricted in their distribution. So for next steps, we're currently working on mapping and quantifying the extent of search effort in historical habitat as well as potential habitat. Then because each species has its own unique ecology, we'll need to factor in detection probabilities. That is the probability of detection for a given species given so much search effort. And finally, we're going to develop a set of Bayesian occupancy models that will formally determine the probability of extirpation, which we're going to frame around IUCN criteria for species extinction or extirpation. And this is really the litmus test for studies aiming to detect biodiversity change in the form of local extinction. So that's a taxon's presumed extinct when exhaustive surveys in known and or expected habitat at appropriate times throughout its historic range have failed to record an individual. Surveys should be over a time frame appropriate to the taxon's life cycle and life form. So by framing our study around these criteria, we'll advance a novel protocol for the detection of species extirpation while testing the limits of concerted community-based efforts to meet these requirements with reasonable degrees of certainty. So yeah, as takeaways from this presentation, the first step to the detection of biodiversity change at the local scale is to develop a system for the synthesis of historical, baseline, and contemporary observational data. Important to note, for different taxa, bumblebees, vascular plants, birds, etc., each are likely to require different approaches to detecting change. Spatial scale matters, of course. Our case study concerns an island, which is a relatively ideal model system for studies in biodiversity change. But depending on the scale of your study area, whether it's open or closed, that is to incoming species, different considerations may apply. And detecting biodiversity change, of course, is only the first step. We care to prevent changes from taking place in some cases. But detecting change in the first instance is a non-trivial task that we need to address in order to monitor biodiversity and prevent its loss. So through this work, we aim to set an example for the role that community science can play in our long-term effort to develop systems for the detection of biodiversity change. The challenge that we've committed to through COP15 is a global challenge, but our results demonstrate that we can make a difference in addressing that challenge at the local level. Through IMERSS, we're actively working with communities throughout the Salish Sea with the goal of increasing community capacity for long-term ecological research. We aim to do this both by setting an example on Galiano, as well as by innovating novel protocols, frameworks, and technologies for the synthesis, visualization, and analysis of biodiversity data. So I welcome you to reach out if you're interested in the open source systems we're developing. It's all available on GitHub. And we can chat about this, of course, in the next stage of this presentation. Thanks so much for your time. I should just acknowledge there's too many names to list, but many organizations have, of course, helped to make this work possible. And again, many thanks to the CDFCP for hosting this event.
Lindsay, I'm just wondering, are there any questions in the chat? We've got one from Pamela, which is, do you want to come off mute and ask it? So I can ask it for you if you want. It's in thinking of growing value of community science, how can curators and organizers of projects help diversify volunteer or community science preferences so that we're capturing a wider range of organisms? Pamela mentioned plants, fungi, and other slow-moving organisms, but many folks focus on organisms they like best, like birds, which mean that some of the more cryptic and less charismatic species aren't getting noticed. So how do you pick up those more sort of secretive species?
Yeah, it's a challenge, and it's interesting because now I'm find myself studying lichen symbioses in relation to biodiversity change, and this is just one of those really cryptic groups that presents lots of challenges for community science. But I think, if you look at the UK, there's like this very long tradition of natural history there, and if you look up, I think I can't remember the Bryophyte Atlas of the British Bryophyte Atlas, I believe. They also have one for lichens. And it's just a testament to when you have the culture, when you have the people for a long enough period of time who are into these things, you can generate amazing, amazing data sets. So I think a lot of this is just sort of a cultural thing, and promoting awareness and interest. So you need to get people, like in a place like Surrey, maybe you could reach out to Terry McIntosh. For example, when it comes to bryophytes, he's a pretty charismatic fellow. He loves to do educational outreach events. So you could do a moss walk with Terry McIntosh and see how many people you can recruit. But no, I think this is a long-term challenge. Through iNaturalist, I've sort of gravitated, or I've found lots of over the last 5 or 6 years in people who study these obscure things, and we've just sort of have formed our little group, and now we're starting to organize and try to really promote that culture. So one thing we're working on, for example, is creating a bryophyte guide for BC. But I think this is a long-term struggle or challenge.
So that's all the questions that we have in the chat for the moment. Is in maybe you can all have a think about some additional questions that we can maybe have a chat about at the end of it. But Dion, do you want to introduce the next speaker?